Sure. So my talk, which is now the second year that I've done it, is part of a course that has actually been a part of CMSC for many years, currently run by Scott Newsom from Hopkins, and it's called Immunology for the Non-Immunologist. And so it's sort of like, I think Dr. Calabrese described it today as like an immunology boot camp to sort of get refreshed on understanding how immune cells are involved in MS and what we can do about it. And this is a complicated field with a lot of things changing. And so I think based on the number of people that have come in the past few years that I've been a part of it, there's a lot to learn. My portion of the talk was really, so Lisa Fox sort of introduces the immune cells and what they do for healthy function. And then I've been sort of the transition to our advanced talk, which this year was from Dr. Calabrese, kind of highlighting treatment development towards novel targets. And my talk is the intermediary, so talking about how peripheral immune cells and the cells that are in the brain and spinal cord can be, their function changes because of the inflammation that happens in MS, and to sort of share with everyone all of the ongoing understanding of how these processes are better being understood so that we can talk about this with our patients. Because the key is that we have so many treatments in MS that target the immune system, but they don't necessarily target fixing the lesions that you're left with and how we diagnose it. And so it's really important to understand how wonderful our medications are, where they're acting on peripheral immune cells, and what we're not getting to, so that we understand what we still need to develop to be able to fix it. It's a really long conversation, which again, we're really lucky in MS care right now to have a long conversation to talk about all the different mechanisms. There's different types of ways that you can treat the injectables, oral medications, infusions, how many side effects you can have from them, how frequently you get them, and how effective we think they are. And so you have to have a conversation with the person sitting in front of you and go through all of these sides of our understanding for why any of them would be a good choice for them or a bad choice for them, to come up with where we're going to start, for example, or what we're going to change to if we need to. And overall, what I talked about today was really understanding that my portion talked about like lymphocytes and monocytes, and how most of our treatments are preventing those activated lymphocytes from entering the CNS or depleting them so that they're not available to do that anymore. And then the second half of my talk was really discussing the targets of the inflammation in the brain and spinal cord, like oligodendrocytes, myelin, the oligodendrocyte precursor cells that can participate in inflammation. It turns out in MS, even though they're the cells that are supposed to replace the lost oligodendrocytes. And then microglia, which are the innate immune cells in the brain, how they can participate, as well as astrocytes, because these cells are responding to using the same signaling functions that we've learned from the immune cells that are circulating in the blood. They're responding and communicating with each other under normal conditions, under healthy conditions, and also in MS. And it's really key to understand all of the mechanisms that are involved in how the cells are interacting with each other, so that we can figure out how to stop that process from happening or reverse it. I think about MS as a two-compartment issue. You have the immune cells that are circulating in the blood, and where they can interact with brain cells and spinal cord cells, or otherwise known as the central nervous system. And so those borders are the blood-brain barrier, at the CSF, and in the meninges. And those are all areas that we can target eventually for therapies to fix the lesions ultimately. But currently, we're sort of outside of the central nervous system in our treatment repertoire. The really key points are understanding, and we're developing the tools and developing the biomarkers to be able to follow how those lesions on a cellular level are changing and how all of those different types of cells are responding, and that will guide the future treatments for repair, whether it be one target or multiple targets, because there's so many cells participating. And that was one of the discussion points actually at the end, where with all of these complex interactions, how do you envision a treatment? And it could be, in the end, a precision immunology or neurology or MS, precision MS treatment, where we figure out what cell is the problem in each person and we can target that one specifically, which would be really cool if we were able to have the markers to do that. We have the tools now to study. This is one of the things that I study in my lab actually, like how do microglia and astrocytes contribute to the process of remyelination? How are they getting in the way? And so with the goal that if we're able to really describe these mechanisms in detail and find the targets that we can manipulate to either reverse the, one of the concepts that we talked about today was not necessarily getting rid of the cells that are causing problems, but change their behavior so they go back to healthy behavior, right, and are able to restore that healthy function. Once we know how to manipulate the cells that way, then we're going to be in a really good place.